Assalamualaikum and Selamat Sejahtera. We are from Group 7, where today we are going to present about centrifuge. So here we have our group member, which is the first one is Nur Umaira Nadia, and me, Nur Ainun. And then we have Nur Hanizati Izati, and Nur Batrisha, Janatul Huda, and our last group member is Rina Shini. So the name of our laboratory instrument for today is centrifugation, where the purpose of the instrument is centrifugation use laboratory technique that use a centrifuge to separate components of a mixture based on the density using centrifugal force. So in biochemistry experiments, centrifugation is important for separating cells, organelles, and biomolecules. It's also important for sample preparation and purification, where it ensures accurate and reliable result.
Next, I will be explaining about principle of operation of a centrifuge. First, a centrifuge operates based on centrifugal force. Based on the animation that I prepare, when the centrifuge is switched on, it spins the samples at a very high speed. We're moving on to the next diagram. Before centrifugation, the sample inside the tube is uniformly mixed, meaning all components are evenly distributed in the liquid. During the high-speed spinning, centrifugal force pushes the particles outward, away from the center of rotation. This causes separation based on density and size. Heavier and denser particles experience a greater force and move faster towards the bottom of the tube. As shown as in the diagram, after centrifugation, these heavier particles settles at the bottom and form a solid layer called the pellet. Meanwhile, lighter components remain in the liquid above the pellet, which is known as supernatant. This principle allows centrifuge to effectively separate biological samples for further analysis in biochemistry laboratories.
So next is I will present about the part of centrifuge. So there are basic and major components. So for the basic components, the central shaft is the part that rotates when the centrifuge is operating. It acts as the main rotating axis and allows spinning motion to occur during centrifugation. Next is the head. The head is connected directly to the central shaft or spindle. Its function is to hold the bucket securely and transmit the rotational motion from the shaft to the sample holder. And another part is the bucket or tube, which used to hold the test tube containing the samples. During spinning, it supports the samples and allows separation to take place under centrifugal force. For the major components, the rotor is the most important component of the centrifuge. It is directly responsible for separation efficiency. Different types of rotors are used depending on the application, such as fixed angle or swing out rotors. The motor is another major component. It determines the maximum speed and stability of the centrifuge. A good motor ensures accurate results by reducing vibration during high-speed rotation. Next is the safety lid with lock system. This prevents the lid from opening while the centrifuge is running. This component is essential for user safety, especially during high-speed operation. And last is the drive shaft, which transfer rotational motion from the motor to the rotor. It ensures smooth, balance, and stable spinning during centrifugation. Next slide. So in conclusion, all these components work together to ensure efficient, accurate, and safe separation of sample in a centrifuge. That's all from me.
So next, I will explain the basic operating procedure of a centrifuge. First, the samples are prepared by placing them into appropriate centrifuge tubes. It is important to make sure the tubes are filled equally and properly labeled to avoid confusion. Next, the tubes must be balanced before centrifugation. This means placing tubes of equal volume or mass directly opposite each other in the rotor. If there is an odd number of samples, a balanced tube filled with water or buffer is used. Balancing is very important to prevent vibration and damage to the centrifuge. After that, the tubes are carefully loaded into the rotor and the centrifuge lid is closed securely to ensure safety during operation. Then the operating parameters are set, including the speed in RPM or RCF, the time, and the temperature if a refrigerated centrifuge is used. Once everything is set, the centrifuge is started and allowed to run until it stops completely. The lid should never be opened while the centrifuge is still running. Finally, after the centrifuge has stopped, the samples are removed carefully. The separated components can be observed as a pellet at the bottom and a supernatant at the top. After use, the centrifuge should be cleaned and switched off. That's all.
So next, I'll be presenting about data output and interpretation. So when interpreting data output from a centrifuge, several key parameters guide our understanding of the separation process. The relative centrifugal force, RCF, measured in xg, reflects the actual force applied to the sample, while the rotational speed, RPM, depends on the rotor radius and influences how quickly components are pulled apart. Time and temperature also play crucial roles, affecting both the efficiency of separation and the stability of the sample. After centrifugation, we observe two main components: the pellet, which contains the denser material settled at the bottom of the tube, and the supernatant, the less dense liquid that remains above. A compact pellet accompanied by a clear supernatant typically indicates successful separation. In contrast, a small or loose pellet or a cloudy supernatant may suggest that the applied force or duration was insufficient. Additionally, an off-center pellet often points to improper balancing of the tubes, which can compromise the accuracy and safety of the procedure. Understanding these outputs helps ensure reliable results and guide adjustment for future runs. That's all for me.
In conclusion, the centrifuge is a fundamental laboratory instrument widely used in biochemistry experiment. It works by spinning samples at high speed to separate substance based on their density. Heavier particles move to the bottom of the tube to form a pellet, while lighter components remain as the supernatant. This allows effective separation of solid from liquid or different cellular components. It is important to use the centrifuge correctly, such as balancing the tube, selecting the appropriate speed and function, as proper operation ensure accurate separation. Overall, the centrifuge play a crucial role in biochemistry experiment to separate the biomolecule, purify the biomolecule, and analyze substance based on density, size, and shape. Proper knowledge of its function and correct usage is essential for producing reliable results and maintaining laboratory safety. That's all from me. Thank you.